The stomach functions as a temporary storage tank for food. This is the first place in the alimentary canal where food spends a few hours being digested. Food typically spends about 3 to 4 hours within the stomach. Here we see that protein digestion will begin. We see that the bolus of food will be converted into a paste-like material that is called chyme. And we also see that the stomach has the ability to expand significantly. An empty stomach has a volume of only 50 milliliters, while a full stomach can expand to 4,000 milliliters in total volume. The stomach has an impressive ability to expand and collapse due to the folds in its mucosa. These folds are referred to as rugae. So let's examine the general anatomy of the stomach. The cardia, or cardial part, is the region that is distal to the esophagus, and it's named for its proximity to the heart. The fundus is a dome-shaped region that's found beneath the diaphragm muscle. The body is the mid portion of the stomach, and the pyloric region is the distal end of the stomach that leads into the small intestine. The pyloric region is shaped like a funnel. So we see that the initial part of the pyloric region, which is called the pyloric antrum, narrows into the pyloric canal. The pyloric sphincter is a valve that is between the pyloric region and the duodenum. And the pyloric sphincter controls the flow of chyme from the stomach into the duodenum. The lesser curvature is found on the medial side of the stomach, so that is, it is the side that is closest to the midline, while the greater curvature is on the lateral side of the stomach. The vascularization of the stomach primarily nourishes the tissues of the stomach, so we see that there are very little nutrients that are actually absorbed by the stomach. We do see some water and some substances like alcohols are absorbed, as things like alcohols can easily move across plasma membranes, and so it makes sense that they can be absorbed within the stomach. The major arteries that serve the stomach are the gastric arteries and the splenic arteries, both of which branch off of the celiac trunk. We see that the celiac trunk is a small branch that emerges from the abdominal aorta. The major vein that drains the stomach into the liver is the hepatic portal vein. The autonomic nervous system controls gastric muscle contractions as well as gastric secretions from gastric glands. We see that the sympathetic division of the autonomic nervous system tends to inhibit gastric secretions as well as inhibiting muscular contractions. A bundle of nerves that is called the celiac plexus is responsible for administering sympathetic control to the stomach. The parasympathetic division typically promotes gastric secretions and also promotes muscular contractions of the stomach. Neurons within the vagus nerve exert parasympathetic control. In the stomach, we see that both the muscularis externa and the mucosa are modified. Typically, we see that the muscularis externa of most of the alimentary canal has 2 layers, but it has 3 layers in the stomach. We see the longitudinal layer and the circular layer, which are the 2 primary layers of the muscularis externa of most of the alimentary canal. But we see that the stomach has a 3rd layer, that's called the oblique layer. So the longitudinal layer is superficial, the circular layer is the middle layer, and the oblique layer is the deepest layer. These 3 layers assist with pummeling food in order to break it down into chyme. They also help to push the chyme toward the small intestine. The mucosa of the stomach is also modified, that is, compared to other mucosa cells that we've seen so far. The epithelium within the stomach is a simple columnar epithelium, which isn't particularly unusual for a place like the stomach, but this epithelia is modified to produce an alkaline mucus that traps bicarbonate. The bicarbonate helps to build an alkaline barrier that will neutralize any acids that are approaching or touching the walls of the stomach. This prevents the acids inside the stomach lumen from damaging the stomach mucosa. So notice that the spelling here for mucus is different because mucus refers to the cells of a membrane. So these are cells that are specifically producing the mucus proteins that are composing that alkaline mucus that we see lining the stomach mucosa. The simple columnar epithelial cells also have abundant tight junctions that function to seal the spaces between adjacent epithelial cells. This helps to prevent gastric juice as well as acids from getting between the cells and seeping into the deeper tissue layers. The stomach mucosa is lined with many small pits that function as gastric glands. There are a few different types of cells within the gastric glands that produce different substances that contribute to the formation of this gastric juice. We see that approximately 3,000 milliliters of gastric juice is produced per day. One of the cell types within the gastric glands is the parietal cell, and parietal cells function to secrete two substances. The first substance is hydrochloric acid, or HCl. Hydrochloric acid has several functions. The first is that it denatures protein, so that is, it essentially breaks down the three-dimensional shape of the proteins that are entering the stomach and aids in the enzymes that are produced in the stomach to digest them much more easily. We see that hydrochloric acid generally has a pH between 1.5 and 3.5. Typical pH of the stomach is about 2. We also see that hydrochloric acid functions to activate pepsin. So the pepsin is the enzyme that's found within the stomach that digests proteins. We also see that hydrochloric acid functions to destroy plant cell walls. So plant cells have a tough cellular wall that is made out of a carbohydrate called cellulose. We are unable to digest cellulose, but we see that hydrochloric acid does help to punch holes in the cellulose so that our bodies can access the nutrients that are inside of plant cells. We see that hydrochloric acid also functions to destroy any bacteria, or a majority of bacteria that have made their way from the mouth or the esophagus into the stomach. So let's briefly look at the mechanism of hydrochloric acid production. The parietal cells, which are shown in blue in this diagram, convert carbon dioxide into bicarbonate using an enzyme called carbonic anhydrase. We saw the same enzyme converting carbon dioxide into bicarbonate in the red blood cells. So in this process, we see that hydrogen ions, or H+, are created, and they are pumped into the stomach lumen. Bicarbonate is then pumped out of the parietal cell, and it moves into the bloodstream. This is a phenomenon known as the alkaline tide. Because bicarbonate is a negatively charged substance that was pumped into the tissue fluid, we see that a balance of electrical charges must remain consistent within the interstitial or within the tissue fluid. So we see that as bicarbonate is pumped out of the parietal cells, a negatively charged chloride ion, that is a Cl negative ion, is pumped into the parietal cell and then into the stomach lumen. It's in the stomach lumen that this chloride ion then combines with hydrogen ions in order to form hydrochloric acid. So that initial pumping or exchange of bicarbonate for chloride is ensuring that a balance of electrical charges is kept within the interstitial fluid. Parietal cells also secrete a substance called intrinsic factor, which assists in the absorption of vitamin B12 within the small intestine. So vitamin B12 is essential for the proper maturation of erythrocytes, or red blood cells. So without enough vitamin B12 being absorbed, pernicious anemia can result. Another population of cells within the gastric glands are the chief cells. And chief cells function to secrete pepsinogen. And pepsinogen is the inactive form of the enzyme pepsin. So typically enzymes are produced and secreted in their inactive forms so that they do not digest the cells that produce them, particularly for enzymes that digest proteins. Pepsin digests proteins, and we see that the stomach is where protein digestion will begin. The chief cells also secrete lipases, which function in the digestion of lipids. So gastric lipases, although they do digest lipids, are not the major factor that contributes to the digestion of lipids. So lipids are more fully and completely digested within the small intestine, but we see that there is a minor start to the digestion of lipids within the stomach. So we see that the chief cells secrete pepsinogen. Again, that's the inactive form of the enzyme pepsin. And we see that the parietal cells secrete both hydrochloric acid and intrinsic factor. The enteroendocrine cells within the gastric gland secrete a number of hormones, one of which we discussed earlier, which was called gastrin. The primary stimulus for the release of gastrin is partially digested proteins within the stomach. Gastrin actually targets the stomach itself, causing the increased release of HCl, or hydrochloric acid, which then aids in chemical digestion within the stomach. Gastrin also stimulates muscle movements in the small and large intestines in order to help propel food masses once they reach those organs.